Hey guys, welcome to topic 3, genetics as an experimental tool. We're going to go through some of these experiments here really quickly. The big key I want you to take away from this is what these experimental design tools allow you to do and how we would use these for further research, just as a way to dip your toe into some genetic experimentation. So here's our topic outline. There's going to be 5 main sections we're going to talk about, and we're going to go through each of these. As always, here's your topic objectives. Please let me know if you have any questions about these, and we can go through some examples of these in class as well. Alright, so let's get started. So at the beginning of genetic research, we've always, the first thing we were able to do was do a random mutagenesis. Now what we do is we take a normal gene sequence, treat it with DNA damaging agents, creates a whole lot of mutations. Now one of the keys about this is that we would need back a long time or a while ago, we would need an organism that is going to create a lot of offspring and do it really quickly. So we could see what the product of these mutations did. So then we would say, oh, here's a fruit fly with white eyes. All right, let's look and see what this mutation is. And that's how that worked. Nowadays we can do this a little bit quicker. Well, we can apply this technique to human genes and applications through cell culture. We can't do it in an organism fashion because first of all it's not ethical, and second of all we don't produce fast enough or create enough offspring to make it something we can do. But we can do this in cell culture as well, and what this allows us to see is where the different phenotype genetic mutations are going to show up. We can then sequence their genome, find out where the differences are, and so that's how this random mutagenesis works. So it's really a useful technique when we want to see what if we're just looking for a random change to see how this changes things. It's really a shotgun approach to looking for a mutation. So then what we can do with this is we can do a genetic screen, or we can do this with normal organisms as well. What we do is we take cells or the organism or whatever, and we can compare them to each other, and then we'll see how these genetic screens work. So if we are looking for cells that can't handle calcium, we can put them all in there, label them all up and have it set, and then we'll be able to check and see which ones can't handle calcium, and we'll be able to go back to the plate that doesn't have the calcium and use that as our way to move forward. Another option we have, because part of the problem with this is that if we want to study something that is very life dependent, so if we want to look at ribosomes, we can't knock out the ribosome. What we can do is we can make them conditional. So what we do is we change them so that they only don't work at 23 degrees in this case on this picture. And so, or I guess they work at 23 degrees in this picture and they don't work at 36. So what that allows us to do is then we can isolate out the mutation and be able to look at it a little bit more. And so these switches help us do that a little bit more because, for instance, in my research, my enzyme is needed for neonatal development, so we can't turn that off until after the cells have started to divide. And so what we do then is we can turn off that gene that creates that conditional mutation for us that allows us to look at it. Another genetic test that we can do is a complementation test. So what happens sometimes is we have 2 different mutations, and we don't know if that mutation is coming from the same gene, and so these are just different phenotypes of that gene, or if these are 2 different genes that have a similar function. So what we can do is we cross 2 homozygous mutants, and what we do, and then we look to see what their offspring looks like. So if you have these 2 white chickens and you don't know if the white gene or the white color is coming from the same gene, you cross them both, make sure they're homozygous, cross them both, and if the offspring shows color, then you know that those are coming from 2 different genes, because one organism is going to have the wild type genes for one gene, and one organism is going to have the wild type genes for the other organism or for the other gene. And when they cross, the offspring will have the wild type gene for both of them. They'll just be heterozygous. If the mutation is on the same gene, you're going to now have a new phenotype organism, because they will have one mutant gene from mutant type A, one mutant type B gene, and those are the alleles that are going to create this different phenotype. So that's how that works. So make sure you understand how that complementation test works. It's a little bit confusing, but that's how we can tell whether or not these organisms are really, or whether or not these mutations are coming from the same gene or not. Another big thing that we do in genetic testing, this is becoming a very big backbone of research, is single nucleotide polymorphisms. Now, if you remember back to Unit 2, we talked about how you and I vary by about one base out of every 1,000 nucleotide pairs, and these are called single nucleotide polymorphisms. Most of the time, these are silent mutations that haven't caused a problem, obviously, because otherwise we would have some impairments, and these neutral mutations have been passed along, and so we can generally see where these SNPs are. Now, what'll happen is what we can do is we can line these all up and compare across several people to see what these changes mean. So for instance, if we're looking for like some kind of precursor to heart disease, what we can do is take a whole bunch of people with heart disease, sample them, compare and look for an SNP that's similar, and then a whole bunch of healthy people and look for and compare their SNPs to the group with heart disease. And what this can help do is help us narrow down as to where these SNPs are occurring and what SNPs matter. And this may help us to identify the genes that might be involved. So that's what's important about that aspect of it, is that it's one more tool that allows us to look for these larger blocks of or these larger, more complicated diseases. But the single mutations can also help us find really easy genetic disease where we find a single mutation that causes this disease. So that's another aspect of these single nucleotide polymorphisms. So these SNPs can also occur in big chunks known as haplotypes. And what these haplotypes have allowed us to do is allowed us to trace human evolution and human migration patterns. Because as you know from crossing over, and we talked about that in topic 1, is that genes are going to, or every chromosome is going to have a crossing over event. And they have multiple crossing over events. So when these these big haplotype blocks are likely to be mixed up. So the more similar the haplotype blocks are, the more likely these individuals are to be related or at least come from a similar genetic line. And you can see an example here with individual B and individual F. They have a similar haplotype block compared to C, D, E, and A. And so this can help us trace ancestral histories and migrations and help us see how the human population has evolved and moved around. Because these haplotype blocks will constantly be changed through crossing over and other items like that. So I know that this was really quick and brief. I'm not too worried about this topic as much for you guys' understanding, but I do want you to be able to understand what an SNP is and how the haplotypes work, and then what these basic ideas of these genetic tests are. But the big focus for you guys in this chapter is topic 1 and topic 2. If you have any questions, let me know.